Hi guys, welcome back to my channel. This is Dr. Rajeshwar from YR Pharma Tube. In the previous video, we learnt the oxidations involving carbon-oxygen bond. If you did not watch the video, hit the I button on top right of this video to watch it. To watch the other topics of the drug metabolism and medicinal chemistry, click on the links given in the description right below this video. In this lecture, we shall try to understand the oxidation reactions involving a carbon-sulfur bond. At the end of this video, you will be able to understand the oxidation reactions of drug molecules possessing carbon-sulfur bond. These oxidation reactions catalyzed by cytochrome P450 and some are selectively catalyzed by flavin monooxygenase enzyme include number 1 oxidative s-dealkylation, number 2 desulfuration, number 3 s-oxidation, and number 4 dimerization. Sulfur-containing compounds that are susceptible to oxidation include thiols, sulfides, and disulfides. In addition, some of their initial oxidation products can serve as substrates for further oxidation. For example, the oxidation of thiols, also known as sulfhydryl groups, which lead to a sulfenic acid. Sulfenic acids can be oxidized further to sulfinic acids and sulfonic acids, but this is not usually observed in vivo. Depending on the substrate, this pathway is mediated by cytochrome P450 and or flavin-containing monooxygenase enzymes. Another route of oxidation of thiols is to disulfides either directly or by dehydration between a thiol and sulfenic acid. However, the mechanism is not clearly understood. Sulfenic acids are reactive substances, the major reaction being reaction with other thiols to form disulfides. In vitro, this usually leads to disulfides of the drug. However, in vivo of the concentration of biological thiols such as proteins or glutathione is higher than the drug, and therefore the major product is a mixed disulfide between the drug and biological molecules as shown in the figure. The metabolism of sulfides, that is thioethers, is rather straightforward. The first oxidation product of a sulfide is a sulfoxide, and this can be further oxidized to a sulfone. Sulfides can be oxygenated by monooxygenases to sulfoxides and then to sulfones. Here, it is known that the conversion of sulfide to sulfone is indeed a reversible reaction as documented by many examples of reduction of sulfoxides, while the reduction of sulfones has never been found to occur. Thiocarbonyl compounds are also substrates of monooxygenases, forming S-monoxides which are called sulfines and then S-dioxides that are called sulfenes. As a rule, these metabolites cannot be identified as such due to their reactivity. Thus, S-monoxides rearrange to the corresponding carbonyl, that is a ketone, by expelling a sulfur atom. This reaction is known as oxidative desulfuration and occurs in thioamides and thioureas. As for the S-dioxides, they react very rapidly with nucleophiles and particularly with nucleophilic sites in biological macromolecules. This covalent binding results in the formation of adducts of toxicological significance. Such a mechanism is believed to account for the carcinogenicity of a number of thioamides. Types of carbon-sulfur bonds. In drug molecules, 4 main types of oxidations can occur with the functional groups that contain a sulfur atom. They are s-dealkylation, desulfuration, s-oxidation, and dimerization. S-dealkylation. The mechanism of s-dealkylation is similar to N and O dealkylation reactions as discussed in the previous videos, and require that the adjacent carbon atom is attached to a hydrogen atom, that is alpha carbon hydroxylation. This reaction is more likely to occur with smaller alkyl groups than large alkyl groups. This is the most common metabolic reaction for the various sulfur-containing drugs or xenobiotics. For example, in rats, 6-methylthiopurin undergoes oxidative demethylation to form 6-mercaptopurin and formaldehyde. In this reaction, the alkyl group, that is methyl group present at the C6th position in the 6-methylthiopurin is removed via an unstable intermediate, that is alcohol derivative, to form the 6-mercaptopurin. Several phenothiazine derivatives undergo s-dealkylation. For example, thioridazine and thiethylperazine. Thioridazine is an anti-schizophrenic agent and thiethylperazine is only used to prevent nausea and vomiting. Both these agents undergo s-dealkylation at the sulfur atom to form the corresponding dealkylated products, leaving the aldehyde. Similarly, s-demethylation of methityrol and s-debenzylation of 2-benzyl-thio-5-trifluoromethylbenzoic acid also have been reported. In contrast to O and N dealkylation reactions, this metabolic route is not seen very often due to the limited number of drug molecules that contain a sulfur atom, that is, the thiol or thioether groups, and also the competing metabolic S-oxidation processes. Number 2. Desulfuration. Desulfuration is a chemical process in which the sulfur atom is removed or eliminated from a drug molecule. In other words, desulfuration is the oxidative conversion of carbon-sulfur double bonds to the corresponding carbon-oxygen double bond. The carbon-sulfur double bond is also known as the thiocarbonyl or thioketone group, and the carbon-oxygen double bond is commonly a carbonyl group. Thus, desulfuration is simply a process of replacement of sulfur to oxygen atom in these transformations. For example, the carbon-sulfur double bond present at the C2 of thiopental is converted to its corresponding oxygen analog, that is called pentobarbital, containing carbon-oxygen double bond. This clearly indicates that the sulfur atom is eliminated from the C2 position of the thiopental. Desulfuration reaction also occurs in the drugs containing phosphorus and sulfur moiety. For example, desulfuration of parathione leads to the formation of paraoxone, which is the active metabolite responsible for the anticholinesterase activity, wherein the PS double bond in the parathione is converted into P double bond O in the paraoxone metabolite. In a similar manner, quasipalm also undergoes desulfuration to its carbonyl derivative. Similar to the above metabolic transformations, only a limited number of functional groups containing a thiocarbonyl group. Number 3. S-oxidation. Sulfides or thioethers can undergo undergo direct S-oxidation to produce sulfoxides and sulfones. Organosulfur drugs containing carbon-sulfur bond commonly undergo S-oxidation to yield sulfoxide derivatives. S-oxidation reactions are normally catalyzed by flavin-containing monooxygenase enzymes rather than cytochrome P450 enzymes. Thus, sulfoxide drugs and metabolites may be further oxidized to sulfones. Note that in S-oxidation process, the first oxidation product of a sulfide is a sulfoxide, and this can be further oxidized to a sulfone. For example, sulindac, a non-steroidal anti-inflammatory agent, is actually a sulfoxide. It can be reduced to a sulfide or oxidized to a sulfone. The sulfide is more active as a NSAID that is non-steroidal anti-inflammatory drug than the parent drug, but the sulfone is inactive. In the second example, thioridazine contains two sulfur atoms. One is in the ring and the other is in the side chain. Both sulfur atoms are susceptible to S-oxidation. Thioridazine is converted to its corresponding sulfoxide and then to a sulfone derivative by S-oxidation reaction. Oxidation of the 2-methyl-thio group yields the active sulfoxide metabolite, that is mesoridazine, and this sulfoxide metabolite undergoes further oxidation to its corresponding sulfone derivative called sulforidazine. Interestingly, in humans, mesoridazine is twice as potent as antipsychotic agent as thioridazine. S-oxidation is one of the important metabolic reaction of the H2 histamine antagonists cimetidine and metionamide. The corresponding sulfoxide derivatives are the major human urinary metabolites. Sulfoxide drugs and metabolites may be further oxidized to sulfones. The sulfoxide group present in the immunosuppressive agent oxysuron is metabolized to a sulfone moiety. Several phenothiazine derivatives are also metabolized by this oxidative pathway. Phenothiazines can be converted to their S-oxides, that is sulfoxides, and then to sulfones by the cytochrome P450 enzyme. For example, in the S-oxidation of chlorpromazine that is shown in the figure. The sulfoxides may be excreted as urinary metabolites or oxidized to sulfones. In humans, dimethyl sulfoxide, abbreviated as DMSO, is found primarily in the urine as the oxidized product dimethyl sulfone. And number 4, dimerization. In chemistry, dimerization is the process of joining two identical or similar molecular entities by bonds. In this case, particularly in drug molecules that contain sulfhydryl groups, can be oxidized to the corresponding disulfides. Similar to S-oxidation, flavin monooxygenase enzymes normally catalyze this metabolic transformation. Disulfides are usually reduced to the corresponding thiols. An example of this is seen in captopril. Two molecules of captopril are oxidized to form a dimer consisting of a disulfide bond. This particular oxidation is responsible for the relatively short duration of action of the drug captopril. Similar to thioethers, sulfhydryl groups are only seen in a very limited number of drug molecules. This is the list of references that are followed for the lesson. That's all in this video, the oxidations involving a carbon-sulfur bond. In the next lesson, we will discuss about the other oxidation reactions catalyzed by cytochrome P450 enzyme. Till then, never stop learning and never stop watching my videos. Thank you for watching this video.